Two main nucleic acids we're going to be learning about is DNA and RNA and the structural differences between them. Nucleic acids are used for information storage, and that is DNA, and transmission, which is RNA. Okay, so they have some structural differences, like DNA has deoxyribose sugar. Okay, so that oxygen group has been removed, and RNA has a ribose sugar, so it has a hydroxyl group. Now, DNA is a double helix, so it's usually shown as a double helix as well. And a double helix just means that it has two backbones that are connected to each other. It means that DNA has strands twisted around each other, and each strand is made of nucleotides. Okay, so there's a nucleotide, 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 nucleotide. Okay, and remember that it is these phosphate and sugar that forms this backbone of DNA. Okay, and then what's different is these nitrogenous bases. So this is a different nitrogenous base. Okay. Now DNA has two nucleotide strands arranged in a double helix. Now, why we say DNA is antiparallel is because if a strand was parallel, it would be 5' to 3' and 5' to 3'. That means it's parallel. But DNA is anti-parallel. So this is the 5' end, and it will move up to the 3' end. Okay, so one strand runs to the 5' to 3' end in this direction, and the other strand runs in the other opposite direction, but it's complementary to the other strand, but it runs in a different direction. Now, this is called anti-parallel. It means that the two strands run in opposite directions from each other. Now, base pairing in DNA. In base pairing, adenine pairs with thymine. So A and T will always pair with each other, okay? And they form two hydrogen bonds between them. And then cytosine pairs with guanine. So C and G will always go together, and they make three hydrogen bonds between them. So what is important to understand is that in this backbone, okay, we have covalent bonds. And between these nitrogenous bases, we have hydrogen bonds. Okay. Now, RNA. RNA uses uracil instead of thymine. Okay, so in DNA, we have A and C, okay, but in RNA, we have A and U connection. So in RNA, adenine pairs with uracil. So A and U will pair together. And this is where it's different from DNA. Because in DNA, as I said there, it pairs with thymine. Now, DNA and RNA have different sugars. Now, DNA contains a deoxy. So D means to remove, okay? And oxy is the oxygen. So it removed the oxygen, and it's a ribose sugar. So it's a 5-carbon sugar. So DNA is deoxyribose because it removed this oxygen group from its sugar. And RNA is just a ribose sugar because it still has that oxygen. So yeah, that's why we call it deoxyribose, because it doesn't have that oxygen. So the sugar is one structural difference. And then the difference in the bases is that DNA has thymine and RNA has uracil. Okay, so here we have an AT connection and here we have an AU connection. DNA and RNA have different bases. Okay, I think we covered that in enough detail now. So DNA contains thymine, RNA contains uracil. Okay, that is different. And both of them have adenine, cytosine, and guanine. They only have that one difference in the nitrogenous base. So the overall comparison, DNA is deoxyribose sugar, so it doesn't have that oxygen group, and RNA is ribose sugar. The bases are the same except for DNA as thymine and uracil. This is typically double-stranded. This is typically single-stranded. Okay, and the structure is it's an antiparallel. Okay, and this is a single strand that can fold into complex shapes. Okay, and stability, this one is more stable, meaning DNA as a double helix structure, it has a longer shelf life. For example, it is more stable as a molecule, whereas RNA is less stable, and then over time it will break down. And then function, DNA is for storage, long-term storage, and RNA is used to transmit information. It's also important in other cellular processes, and we'll get to that as we progress through the units. So the essential knowledge, the takeaway, is that DNA and RNA are both nucleic acids, which is the polymer. Okay, a nucleotide is the monomer of nucleic acids. DNA is antiparallel and it has a double helix. And in DNA, H pairs with T and C pairs with G. And then these bases pair with each other through hydrogen bonds. Okay, very weak bonds. And then RNA usually has one strand and it uses uracil instead of thymine. And then they're also different in their sugar bases, where DNA has a deoxyribose, meaning that oxygen group has been removed, and RNA has a ribose sugar, so it has that oxygen group. And then the bases are different, and the typical strand structure is double-stranded and single-stranded. If you enjoyed that video, please don't forget to like and subscribe, and can join us on the Skool.com community page where you'll find AP Bio blueprints. And if you take a look inside, you will see that Unit 1 is completely free. See you all there.